첫 번째 세션은 IBM 퀀텀의 Dimitris Alevras 박사님께서 mRNA secondary structure prediction using utility scale quantum computers라는 주제로 온라인 발표를 진행하시겠습니다. Now we will begin the first session. Dr. Dimitris Alevras from IBM Quantum will give an online presentation on the topic mRNA secondary structure prediction using utility-scale quantum computers. Please give a big round of applause. 큰 박수 부탁드립니다. Dimitris, you can start now.
Hello, can you hear me?
Yes.
Okay. Hi, it's my pleasure to be here with you today to share our work on applications of quantum computers to the optimization of mRNA. This work is joint work we do with Moderna, which is a leader in mRNA research. The results of this work have been published in the conference proceedings of the QC24 that took place last year in Montreal. So why mRNA? So mRNA, as probably you all know, is the way genetic information is delivered in our bodies. So basically it's the translator from DNA to the functional molecules that are the proteins. It is used to direct the production of protein in humans. So basically it provides a recipe of how to create the proteins. Recently, in the past decade or so, it has been emphasized and pursued, the research has been emphasized and pursued to expedite, to use mRNA to expedite and optimize drug discovery. And that's due to its high degree of designability that allows you to target, to discover drugs for large populations, as we evidenced in the creation of the COVID vaccine a few years ago, and also for personalized medicine to target particular individuals for their particular disease. However, this high degree of designability comes at the expense of high complexity. Optimizing mRNA is a very difficult combinatorial problem, and the importance of understanding how you sequence, modify, and structure the molecule affects the stability and efficiency. And this has to do not only with the behavior of the molecule in the lab, but also in terms of manufacturability and stability in moving from the manufacturer to the consumer. So let me give you an example of what we mean by mRNA optimization. The secondary structure prediction problem that we are discussing today is part of a bigger problem that's called mRNA optimization. You start with a sequence of amino acids. So each of these letters that you see here, it's an amino acid that can be expressed via this table here to a sequence of nucleotides. And this sequence of nucleotides can fold naturally into these pairings, and that's a stable molecule. These pairings are predefined, and then not all the pairings are possible. So what is known in the field as a codon optimization is basically trying to find which of these three-letter nucleotides here can express each of these amino acids. So these three-letter combinations here are called codons. That's why this problem here is called codon optimization. So going from amino acid to nucleotides, this is the codon optimization problem. We are not concerned with this problem in our study here, but we're concerned with this problem, how given this sequence of nucleotides here, I can go from here to there. And how can I predict this stable structure? This is like I said, a hard combinatorial optimization problem. Classically, there have been many approaches. Of course, there is a mathematical programming approach, which is what we base our research on. There is a dynamic programming approach based on a very clever observation by Nusinov and then extended by Zucker. Basically, Nusinov's algorithm just found the maximum number of pairings, but that was not guaranteeing to have the stability of the molecule. And then Zucker introduced the energies. There are more exotic approaches to the problem, like using stochastic context-free grammars, which is a very interesting approach that attempts to solve the whole optimization problem from amino acid to folding. And of course, there is machine learning trying to find the patterns and genetic algorithms and what have you. In any of these approaches that have been going on for many decades, the success rate is about 72%, which means that we can successfully predict what happens in the lab around 72, 74% of the time. On the so people thought, what if we could do something with quantum? And the approaches basically have been based on solving the problem defined classically in one of these methods here with quantum. So basically starting with a math programming approach or a Hamiltonian that you can build as a QUBO, as a quadratic unconstrained binary optimization problem, you can apply quantum annealing, or there have been gate-based approaches that mostly attempt to solve, have solved the problem with simulations in a very small number of qubits on hardware. Digging a little bit deeper into the structures and what we are dealing with. So this is a sequence of an RNA molecule. You start at this point here, and you end at this point here, and the sequence goes like that. So as you can see, it can fold in many complex ways here. And they have identified different substructures that are stabilize or destabilize the molecule. So to start with, if you want this chain here to fold, you need this part, this blue part that's called the hairpin, to have a minimum length, because if it was only one, it would be physically capable of folding. So there is a minimum hairpin length involved as a constraint. And then we know that consecutive pairings are stabilizers. So you want to have long chains of pairs here. As you can see in this upper right picture here, consecutive pairs have like negative free energy, while loops, like the bulge loop you see here, the multi-loop you see here, and the internal loop you see here, as well as the hairpin, all of these are destabilizing forces. Some of them necessary, but then they reduce the stability of the molecule. Finally, you have this structure here that's called a pseudoknot, and it has been proven theoretically that if you model this structure into your problem, this makes it NP-hard. In our initial study, we ignore the loops and the pseudoknots, and we consider all the other constraints plus some non-crossing and some dangling. Dangling constraints would be like things that are falling, that are hanging over the chain here and here. As I said, we are focusing on the math programming formulation to get a quadratic unconstrained binary optimization problem. And there are basically three ways to model this problem. One is to identify your variables as pairs. So for example, you can see here in the green square, the pair is xij. So I have taken a small snapshot here. So you have a chain that goes from I, sorry about that, goes from I to I plus 1, I plus 2, folds like that, and continues down here. So if I want to model this, I model xij as one variable, xi plus 1, j plus j minus 1 as another variable, etc. That's the pairing formulation. Then there is a stacks formulation that basically tells you, okay, to model these three pairings here, I say I start at I end at j, and my stack has a length 3. So this will model this whole structure here. Of course, you could say I can start at I, end at j, and my stack has a length 2, in which case you model this thing here. So all these have to be generated as variables. Finally, there's a quartet formulation that you can see here in the red box. And basically, if I wanted to model it here, I would model with these three structure, I will model with two variables. One variable would be these two pairs, and the other would be these two pairs. So basically it says I, j, I plus 1, j minus 1, this whole thing is a variable. So if this variable is 1, these pairings must be done. So basically, defining my variables in any of these ways and having some energy data, and also being able to identify substructures as constraints or with penalties, I can model this and try to find the most stable structure. In our study here, we chose to go with a stack of the quartet formulation. So basically, our variables are x, I, j, I plus 1, j minus 1, which means that if I is paired with j and I plus 1 is paired with j minus 1, this variable is 1, otherwise 0. And you can formulate this by taking a minimization. You minimize the free energy, which is this. And then you take some constraints that reward consecutive quartets and penalize some structures that are not really desirable as a dangling pairs. Also, we consider these constraints here that basically tell you that the two crossing quartets cannot be both selected. This can be also modeled on the objective function. The reason we have it here on the classical model is that classical optimizers, exact optimizers like Cplex work much better if you have these constraints here rather than the objective function. But the fact that these here are two binary variables that says not both can be one, basically can be easily modeled as I penalize the product being 1. So I want this product to be 0. So by just doing this substitution from this constraint to this constraint and keep the rest the same, I end up with this formulation here, which is a quadratic because I have quadratic terms here and there and here. Minimization, it's unconstrained. I don't have any constraint, it's binary. Now you will notice that there is a difference between the variables I use here and the variables you see here. You can think of QK as a sequential, the sequential indexed variable of this form. That was the mapping phase of my problem. So I know how to take my problem and model it and have a Hamiltonian. The next step was to figure out on a real machine, like on a device, how to select the qubits. So what we follow the paper by McKay and all here that basically takes a layer fidelity benchmarking approach. Basically, you create layers of these joint sets of qubits. In this case, we have three layers: the blue, the red, and the green. And basically, they cover the whole all your qubits, all the neighborings. And then we apply this simple circuit here, which is a swap gate. We have a Hadamard gate here to put this in superposition, you do the swap, and then you put another Hadamard here, and you know what the result should be. So basically, what we do is we execute this, and then we measure the fidelity of the ideal bit string that we should expect to come to receive at the end after measurement with the obtained one. And this gives you fidelity. As you can see, these numbers here is couplings, like pairs of qubits. And basically you see that they are ordered by higher to lower fidelity. So we put a threshold of 85%. So if a pairing is below 85% fidelity, we didn't consider it. This way, we create a 100-qubit chain that exclude the qubits that have low fidelity. And in this diagram here, that is one of our Eagle processors, if you see, I mean, for those familiar, you will notice, first of all, that it's rotated minus 90 degrees. But like, if you follow the path here of the dark colored circles, which are the high fidelity ones, you will, in the colors on the links, indicate which couplings come from, the blue, the red, or the green. You will see that there is a clear a chain from qubit 27 to qubit 37 that contains 100 qubits. And that tells us which qubits to use from the device. That was the optimizing our the device for the runs that we want to make. And then what we are going to run. We chose to use the CVAR VQA, variational quantum algorithm. Basically, variational quantum algorithm is that it indicates that you have an ansatz which is parameterized, and we start with a very simple two local ansatz here, which is shown here. This is a two local ansatz on 3 qubits, and then we repeat this p time, so you have p layers. The entanglement is these control Z gates that you do here, and the one we chose was double linear. So basically, it goes 1 to 2, 3 to 4, and so on, and then the other one goes 2 to 3, 4 to 5, etc. So this is in that sense, with that scheme, all the qubits are entangled, and run this circuit through the quantum computer. You get a sample distribution of samples. You calculate the expectation. Now the expectation is calculated classically. In our case, we don't use the estimator, but rather than the sampler. And in addition, we're using a CVAR approach. CVAR basically is conditional value at risk. It's a method that's being extensively used in finance, and basically says you don't want to consider the expectation of your whole distribution, but you specify a parameter, let's say alpha 10%, and you say I will consider the 10% lower end of my distribution because that's the highest quality solutions that I have. As an external optimizer, we use an FT optimizer, which is remarkably stable under noise in our experiments. The NFT optimizer has the feature that optimizes one parameter at a time. So you have pretty much control of how many iterations you run, because you can run, if your circuit has like 100 parameters, you will run 100 iterations for one epoch, and then you can specify how many epochs you want, so you have control over your value, over your iterations. These runs were done last year when sampler version 1 was there. Since then, this has been deprecated, but that's what we used with optimization level 3. As an error suppression mitigation, we used dynamical decoupling, and also we tried M3, which eventually we ended up not using because it gave us more overkill than value for our particular problem. And here are the characteristics of the circuits that we ran. So we ran the smaller sequences, 26, 40, and 50 qubits. We ran them on Brisbane and Osaka, which are Eagle processors, and the 80 qubit we ran on IBM Torino, which is a Heron processor. And you see the qubit, the circuit depth, and the gates. The ECR gates are native in these Eagles, and the control Z gates are native in Torino. So that was our planning for execution, and then post-processing phase is pretty simple for us. So we have a bit string that's the best bit string we found with our algorithm. We convert this bit string to this construct here that's called the dot bracket. if you notice, the dimension of this is not the same with that. This is the dimension in the dimension of the number of variables that we have, the quartets that we created, and these correspond to each nucleotide. So basically this dot tells me that the nucleotide is not paired. So this C, you see the first nucleotide C here is not paired, but the second one is paired. And there is a unique way to go from here to this, and we're using the Forna server, which is a web service that Vienna RNA in the University of Vienna in Austria provides, and then you can provide this input and you generate the graphic so you can see how the folding works. So that's the whole setup that we have. We map the power problem to a Hamiltonian. We optimize our circuit. We define how we're going to execute and how we're going to process our results. And then let me talk about some results here. So classically, we solved this with simplex. We generated 10,000 sequences of lengths 15 to 60 nucleotides. Simplex had no problem solving those, but you can see that the time it took to solve those started increasing exponentially. And even though our model didn't include all the structures, it achieved like 64% accuracy in the solution. So basically it solved 64% of the solutions to optimality, to the optimal solution known from the classical software they use in biology. Then we ran our results on simulator. We ran our experiments on simulator, and here are the results. So what we did, we took sequences of 10, 15, up to 40 qubits, and we ran each of these 10 times. And for each of these 10 runs, we found the optimal solution in at least one time. And this chart here shows you the probability of success. So when you had 10 qubits, we were always finding the optimal, and we were pretty good at 15, 20 qubits, and then we started getting lower success probabilities, although sometimes we get pretty high mean values. What are some observations from running on a simulator? And when I say simulator here, we use the matrix product state simulator, MPS. So first, the whole scheme is very sensitive to alpha value, which is the parameter of the CVAR. The second is that increasing the P layers gives you better results in the simulator, but as we will discuss, as you can imagine, in the hardware that increases the depth, so the performance deteriorates. Then that's common for like all variational schemes. The last 2 points, it's the starting point sensitive, and they have large flat areas in the landscape. So this is something that can give you to barren plateaus and be stuck in a local optimum that you cannot escape. And this is also a mechanism to escape this optimum, because if you start from a better point, you will not get trapped there. This has been observed by other researchers too, that the particular formulations have these flat areas. So this was encouraging, like at least we hit the optimal solution sometimes. So we decided to run this on hardware. And this slide here is split into two. With the left side is the runs on the Eagle processor for 26, 14, 50 qubit problems, which correspond to 25, 30, and 30 nucleotides. These were on the Eagle processor of Brisbane or Osaka, with one layer in the ansage and 0 to 5 as an alpha. And we used the optimal solution in all three on hardware, with a probability of success, if you draw this line, about 40%. And here it tells you how many times you run. We ran 8 trials here, 14 here, and 15 there. And the number of slots of each experiment was there. That was good, so we moved to solve a bigger problem. Due to runtime limitations, though, we decided to do the training offline. What does this mean? We set our alpha to 0.1, and we used 2 layers. Then we run the simulator to obtain the optimal theta values, the optimal parameter values. And then using the optimal parameter values, we started one iteration on the Heron processor on IBM Torino. And we're able to to get the solution for the 80 qubit sequence, which corresponds to 42 nucleotide length. As you can see from this picture here, we not only we found the optimal solution, but we sampled it with the highest probability of the other solution. So that was really a good experiment. And these are different solutions that we obtained for the 42 nucleotide sequence. And one thing I want to point out here is you see how different your configurations can be in these three here. You have almost the same energy, and how you can see how different the pairings are. So this indicates the problem with the flat areas that I was talking about. Also, you can see how even if you are not optimal, a second, like a close to optimal solution is very close to the real optimal solution in terms of folding. So to summarize, since I'm almost out of time, we set out to examine the feasibility of attacking this problem with a quantum computer, and we found that we can get useful results with minimum mitigation and suppression on real hardware for much larger nucleotide sequences that have ever been tried. Now, in an upcoming paper that should be out this month or the next, we will show results that we exceeded like 100 plus qubit runs. We reached the maximum 156 qubits on the Marrakech, and that corresponds to 60 plus nucleotide sequences. And also we examined ways to reduce the qubit requirement. So basically make a tighter formulation. Observing our solutions, we found that our solutions have very low Hamming weight, so that indicates like a redundancy. This doesn't, sorry, I lost my, so this doesn't have to be quantum. This tightening of the formulation can be on the classical side. Also, our research indicates that we have to use more complex answers and that get more deeper circuits, but they give us better sampling, and also the optimization techniques, the available optimizers, external optimizers for the variational scheme need to be more efficient than what we have right now. So this is an open area of research.